Welcome back to G Fun Facts Online. Today we are stripping away the common myths surrounding attention deficit hyperactivity disorder, or ADHD, to reveal a paradigm shift occurring right now in modern neuroscience. For decades, the public and clinical understanding of ADHD was dominated by a single molecule, dopamine. The narrative was simple and mechanical. If the brain is a car, the ADHD brain was simply running low on fuel. The logical solution, therefore, was to fill the tank using stimulant medications like methylphenidate or amphetamines. But modern imaging and network analysis have popped the hood on that car, and what we found is a far more complex engine. We now understand that ADHD is not merely a chemical deficit, it is a network disorder. It is a condition defined by connectivity, specifically the efficiency, or lack thereof, of the electrical highways linking the impulse control centers of the prefrontal cortex to the deeper, more primal regions of the brain. This realization has sparked a therapeutic evolution, a debate between the established titan of chemical stimulation and the rising challengers of electrical and neuroplastic therapies. The question we are exploring today is no longer just how we treat symptoms, but how we physically change the brain. We are looking at the mechanisms of neuroplasticity versus stimulation. To understand this new frontier, we have to start by re-examining the old guard with a fresh scientific lens. We need to talk about what stimulant medications actually do, because the mechanism is far more elegant than just speeding up a slow brain. In the neurochemistry of an ADHD brain, neurons often fire with excessive background noise. This is the physiological basis of distraction. Signals get lost in the static. When a person takes a medication like methylphenidate, known commercially as Ritalin, or amphetamines like Adderall, these compounds act by blocking the transporter proteins that typically vacuum up dopamine and norepinephrine after a neuron fires. By inhibiting this reuptake process, the medication floods the synaptic cleft, the microscopic gap between neurons, with neurotransmitters. But here is the nuance. You should not think of this as adding fuel. You should think of it as turning up the signal strength on a radio. The medication increases the contrast between the signal and the noise. Suddenly, the neural command to focus on a spreadsheet becomes significantly louder than the background static of noticing a bird outside the window or hearing a hum from the refrigerator. There is also a fascinating, often overlooked aspect of medication: the potential for neuroprotection. Critics often label medication as a band-aid, implying it is only effective while active in the bloodstream. While it is true that the acute focus effects wear off in hours, recent long-term studies suggest a deeper benefit rooted in Hebbian learning. In neuroscience, there is a famous axiom: neurons that fire together wire together. By artificially holding the brain in a highly focused, synchronized state, medication allows neural networks to fire in a rhythmic, organized pattern. It essentially forces the brain to practice being focused. This repetition may preserve gray matter volume in the prefrontal cortex that might otherwise atrophy from disuse. However, we must be clear about the limitation here. Medication is largely passive. It creates the optimal chemical conditions for focus, but it does not necessarily teach the brain how to recruit those networks without chemical assistance. Enter the challengers: non-invasive brain stimulation, or NIBS. These therapies represent a fundamental shift because they do not add chemicals to the system. They speak the brain's native language, which is electricity. The two heavyweights in this arena are transcranial magnetic stimulation, or TMS, and transcranial direct current stimulation, or tDCS. Let's break down TMS first. You can think of TMS as a magnetic mallet. It uses a magnetic coil placed against the scalp to deliver focused magnetic pulses through the skull. These pulses induce small electrical currents in specific target areas, typically the dorsolateral prefrontal cortex. This region is essentially the CEO of the brain, responsible for executive planning and inhibition. The mechanism here relies on long-term potentiation. By delivering high-frequency pulses, usually at 10 Hz or higher, TMS excites the neurons, forcing them to fire repeatedly. This bombardment signals the brain that this specific pathway is crucial. In response, the brain triggers the release of brain-derived neurotrophic factor, or BDNF. BDNF is a critical protein that acts like fertilizer for the brain. It encourages the growth of new dendritic spines, which are the physical connections between neurons. The goal of TMS is to physically strengthen the focus circuit through this growth. On the other hand, we have tDCS, or transcranial direct current stimulation. If TMS is a mallet, tDCS is a gentle nudge. It applies a very weak, constant electrical current, usually between 1 and 2 milliamps, to the scalp. The mechanism here is distinct, because tDCS does not force neurons to fire. Instead, it changes the firing threshold. This creates a state of plasticity. A nodal stimulation, which uses a positive charge, lowers the threshold, making neurons more likely to fire when you attempt to focus. Cathedral stimulation, using a negative charge, raises the threshold, helping to quiet down hyperactive areas. The key to tDCS is that it is state-dependent. Because it only primes the neurons, It is most effective when combined with active cognitive training. If a patient performs a working memory task while receiving a nodal tDCS, they are effectively digging a deeper neurological groove than they would with the task alone. This brings us to the showdown between these modalities, the difference between state changes and trait changes. This is the conceptual core of modern ADHD therapy. Medication provides a powerful state change. It instantly corrects the neurochemical deficit. A study comparing a single dose of methylphenidate against a single session of tDCS found that medication won by a landslide for immediate symptom relief. It is like wearing glasses. You see perfectly while they are on, but your eyes have not physically improved. Stimulation therapies, conversely, aim for a trait change. They are playing the long game. A single session of TMS or tDCS does very little, but over 20 to 30 sessions, the goal is to induce structural plasticity. By repeatedly forcing the focus network to activate, these therapies attempt to physically thicken the neural connections. It is akin to physical therapy or going to the gym. You do not go once and expect to be strong. You go repeatedly to tear down muscle fibers, so they rebuild stronger. Stimulation therapies attempt to build muscle in the executive control centers of the brain. If successful, the benefits persist long after the treatment stops, constituting a true rewiring. However, we cannot ignore the third player in this equation, endogenous neuroplasticity, or the plasticity generated by the body itself. Research into lifestyle neuroplasticity is revealing mechanisms that rival clinical interventions. New studies have highlighted a molecule called irisin. This is a myokine released by muscles during high-intensity exercise. Crucially, irisin crosses the blood-brain barrier and triggers the release of BDNF in the brain. This provides a rigorous biological explanation for why exercise is so often cited as a critical component of ADHD management. It is not just about burning off excess energy, it is about chemically priming the brain for plasticity. Alongside exercise, we have neurofeedback. This acts as a bridge between the biological and the psychological. Neurofeedback uses a brain-computer interface to show the patient their own brainwaves in real time. For example, a movie might play on a screen only when the patient suppresses their daydreaming theta waves and boosts their focused beta waves. The mechanism here is operant conditioning. Unlike passive medication or external electrical stimulation, neurofeedback forces the patient to learn how to self-regulate. It trains the driver, not just the car. So where's this all heading? The binary choice of pills versus pulses is rapidly becoming obsolete. The future of ADHD therapy is combinatorial psychiatry. We are moving toward protocols where a patient might take a low dose of a stimulant to normalize the chemical environment and lower the background noise. Simultaneously, they might undergo tDCS stimulation to prime the prefrontal cortex, sensitizing the neurons. And while that stimulation is active, they engage in computerized cognitive training to fire those specific circuits. This Swiss Army knife approach leverages the strengths of all mechanisms, the immediate clarity of chemistry, the priming capability of electricity, and the specificity of active learning. For the millions living with ADHD, the landscape is expanding. We are moving away from a model of mere maintenance, keeping symptoms at bay day by day, toward a model of mastery and modification. Whether through the precise magnetic pulses of TMS, the gentle current of tDCS, or the targeted chemistry of modern pharmaceuticals, the goal remains the same: to harness the brain's incredible, innate ability to change itself. We are no longer just filling the tank, we are rebuilding the engine, one synapse at a time. Thank you for listening to this deep dive on G Fun Facts Online.